Good morning and a warm welcome to today's Zeiss webinar. Today we will be talking about the most versatile microscope for life sciences. My name is Johanna Neu and I will be your host today. Our speaker joining me virtually today is Chris Power. Chris has been with Zeiss for over 20 years, starting at product management for confocal microscopy before he moved into research and development of 3D imaging. And Chris has now taken over a new role as product and application specialist for LSM systems in Europe. And now, without any further delay, I would like to hand over to Chris. So Chris, the stage is yours.
Good morning. Let me just check that my screen is showing.
Yeah, we can see your screen. Looks good.
Okay. So this morning I've kind of been asked to give an interesting talk. Of course, they're all interesting, but the interesting dilemma of how do you make a talk which appeals to both new people who are new to confocal and experienced veterans. So I hope I can do that over the next 40 minutes or so. I want to concentrate on the history of confocal, how these machines have become the workhorse in the field they are today, to go through how we've improved these systems over time, and then to paint out some of the vision we have at Zeiss of where these systems are going and how they can continue to be the workhorses in labs in the future. If I start, let me check. Here we go. So yeah, as I mentioned, the basics of confocal started long ago as a routine imaging system. And over time, these have become more and more powerful. To start any talk on confocal, I will briefly explain laser scanning confocal for those of you who aren't familiar. Laser scanning confocal, I always think of as really just being as simple as the name. So laser scanning confocal, there are three elements to it. The first is the laser, where we shine a laser in, we use a beam splitter, which reflects only the laser to the sample, and then we collect the returning light onto a detector, typically a photon multiplying tube. The reason why we use lasers to do this is really twofold. One is that they're monochromatic, so you get very specific excitation of fluorophores. If you think about the wide field world where we have mercury, that wasn't necessarily so. Okay, with LEDs coming, that's now changing. But yeah, certainly lasers have allowed selection of dyes much easier in the past. You also have the power to focus light down to a single spot. So that's the laser element. The scanning element refers to how you collect the image. So if you think about a wide field system, you excite and you collect the emission simultaneously for every pixel in the field of view. In the case of confocal, you don't do that. You scan, which is where you move that laser spot over a single pixel at a time and you detect a single pixel at a time. Now, that lets you build up an image of arbitrary pixel size and dimensions so you can adjust your sample. But interestingly, it means that from an optics perspective, you can have a single spot on the return. And that's where the third word comes in, which is confocal. So we have this challenge about how do you only get the light from the focal plane? Because everything out of focus isn't helping your situation, isn't helping the analysis of the sample. And this problem was luckily solved for us by a gentleman called Marvin Minsky. Now, back in 1956 in Harvard, Marvin sat down, thought about the problem of out of focus light, and presented this idea in a patent. And this is the idea of having a pinhole in between the light source and the sample in order to create a focused spot. We've replaced that since with lasers, but also to have a pinhole on the return path. Now, that idea in 56 was maybe a little bit ahead of its time, but by 1984, that was commercialized in the confocals. So going back to our light path, what you see here is a conjugate focal plane. And what this means is any light which is in focus will pass through that pinhole. Any light which is out of focus hits the pinhole and gets blocked. And really the sectioning, so the optical sectioning within your microscope is determined by a number of parameters, but mainly the numerical aperture of the lens that you're using, the wavelength of light, and the size of that pinhole. So by changing the size of the pinhole, we can change things like the sectioning strength. And a couple of simple graphs. I'll try and keep the maths basic in the talk today, but most people know you set a pinhole to 1 area unit, so you just capture the 1 volume. And the reason for that is it has a reasonable relationship on the section thickness, but also when you think about the resolution and the sensitivity of the system, if we set the systems to 1 area unit, marked by the red line here, you get most of the light to come through the system. You can close it down to improve the resolution, but then you begin to throw away a lot of the light. So since Minsky's idea was commercialized in 1984, confocals really have moved on to become the dominant systems used in labs today. And that's because they're just very, very versatile in terms of optical sectioning. Here, hopefully, if my bandwidth is holding up, you'll be able to see some examples from a whole host of applications. And this out of focus light rejection works extremely well, not just on cells or monolayers, but on samples of really quite thick. So here we see the central nervous system of a cockroach, which is sectioned, and then here we're rendering it in 3D using Airyscan. So by the 90s, confocals had already become dominant as a way of removing out of focus light and of collecting these beautiful pictures we see. But since then, the systems have evolved to become a bit more sophisticated. One way in which they've done that is by really taking to heart the challenge of spectral. So I say the challenge of spectral. This is the idea that you might have multiple wavelengths of light coming out of your sample and you want to detect them all simultaneously, which is something we do in the 980. And the reason for that is fluorescent probes since around about 2002, 2003, where there was an explosion in colors, have become a main tool used in labs today. If I give you an example here, this is 4 different fluorescent labels in a plant cell, which also has chlorophyll autofluorescence. We can now image that and we can separate out the different colors. So you don't have to worry about endogenous autofluorescence. And this technique gets really, really powerful. If I show you the next example here, this was a paper where they were trying to look at fungus and also a labeling which was incredibly weak relative to the autofluorescence. So here they were looking at basically the green and the red. The magenta was overpowering. Using autofluorescence, we can pull signals out. We can pull out signals which are maybe 1/10 of the intensity of the autofluorescence, and then we can digitally remove the autofluorescence. So spectral imaging has become a way of looking at exactly what you need to see. And it's also so powerful, you can multiplex. So by multiplex, it's used in many terms. In this case, I'm using it to refer to having many, many dyes within one sample. This is an example labeling of cells where there were 10 colors put into a sample at the same time, and using spectral imaging, we're able to image all of those and pull them all apart. Of course, display becomes a bit tricky because of the limitation of the human eye, but we can see this. And then confocals also offer other abilities beyond imaging. So an example here would be fluorescence experiments such as photoactivation, photo bleaching, where you're looking at dynamics within your sample. So here you might be saying, how many molecules do I have? How fast are they moving? So within the confocals, we can take a series of images, a time series. We can then apply high laser power, we can bleach a sample, and we can see how quickly that recovers. There are a whole bunch of variants of this. So FRAP is the most commonly known one, but FLIP and iFlip and photo conversion experiments, but they're all based on the same idea, which is using a laser to convert and change some of the molecules within the field of view and to see what happens. So this is an example with Qaeda, which is a green photoconvertible fluorophore, which changes red when you excite it. Another thing which came to confocals is FLIM. So this is fluorescence lifetime imaging. Here you can use a pulse laser and a photon counting detector. So either the gallium arsenide PMTs with the system or some other variant of detector. You fire a pulse laser source, you look at when the light comes back, and you can plot the decay curve. And by doing that, you can pick apart things like environmental change, so be it pH or oxygen or ion indicators. You can look at changes in lifetime when two molecules are very close to each other, which is done in FRET experiments. And you can look at things like disease markers where disease tissue might exhibit a different lifetime. Now, that's okay. In terms of telling you what has changed, it doesn't necessarily tell you what there is in a sample. And that's where another technique is very useful in the form of fluorescence correlation spectroscopy. So in brief, you, instead of imaging your sample, you park your lens, you have your confocal volume just stay in an area in your sample, and you look at the molecules moving in and out. If you plot that over time, you can run a correlation. So you look at the typical transit time of molecules within the volume, and you can work out interesting things such as the concentration of the molecules in your sample, how quickly they diffuse, and even the type of movement. So you can tell whether they're bound, unbound in a particular way. So just an example of that here, this was a group looking at transcription, put my teeth back in, transcription factors within an embryo, and they could see how those varied using FCS. Now that was powerful enough. People then moved on to doing fluorescence cross-correlation spectroscopy. This is where you don't do FCS with one color, but you do it with two simultaneously. So you can look at the relative concentration and diffusion of two molecules, and you can work out how in different areas of the cell, these two molecules interact in different ways. And if you take that to the extreme with the architecture of the 980, there are some groups now doing that with up to 6 colors simultaneously and taking all of those 6 colors onto the quasar detector and picking apart the correlations. So you can see how over time confocals have gone from being an imaging system to being much more than that, a powerful analytical tool where you can get real numbers out about what's going on in your system. So FCS is very powerful. There is the kind of old joke that a picture's worth 1,000 words and 1,000 pictures is worth a number. This technique is powerful for a single pixel, but you might want to apply it across the whole image. So that's where another technique comes in called raster scanning image correlation spectroscopy, which is where you scan an image multiple times, you apply a correlation similar to the RICS correlation, and you can plot out maps of how many molecules and how quickly they're moving within your sample. So I appreciate that's a bit whistle stop, but that's a brief overview of some of the broad spectrum of things that you can do with a confocal. If I come back to more basic things in terms of imaging, confocal doesn't live on its own. There are other optical sectioning techniques on the market. I list a few of them here, turf, wide field spinning disk, structured illumination, etc. Now you can prioritize these relative to each other in terms of a feature like depth penetration. You could do it in terms of speed, how well they separate out of focus light. And over time, there are more and more of these systems. I think the last time I counted, there's about 40 that are sold today. But for simplicity's sake, I'm just highlighting a few of the more common ones here. What I'd like to point out on this slide is there is no one system that's better at some. If you want depth penetration, you probably want multi-photon, but that might not be the best system for pure speed. But confocal, so single point scanning, or multi-photon, which I guess is a variant of it, always scores very highly in terms of these metrics. And the combination of that plus all of the features I've just pointed out, all of the flexibility of the system, is why confocals are very, very dominant. In terms of the optical sectioning market, even though there are 30 or 40 systems to choose from, confocal remains well over 50% of the system sold. And that's just a slide showing, you know, kind of the power of the system. It isn't just an imaging system. Now, as good as these systems are, there is always a strive to make them better, to make them a better system to do more and more research. How do people want them to get better? Well, here was a survey from a few years ago where we asked people what they wanted to see. And of course, there are common themes here. There is the triangle of speed, resolution, and sensitivity, which is a bit cliched in these talks. So I've just left it as a bar chart rather than a triangle, but they're the three things that we always strive towards. And they're not necessarily independent. On the right, I have an example of telomere replication where the group are trying to look at how many green and red spots there are. If we zoom in, they struggle to see what's going on. If you improve the signal to noise, you also improve the resolution. So these factors aren't entirely independent. And if you can improve the system in some ways, you can improve it for all of these. Another very strong motivation in trying to improve the confocals is gentle imaging. So everyone's known about photo bleaching for years, in that if you put too much light in, you will bleach your sample and you won't be able to see it anymore. But phototoxicity is also a concern where even if your sample is very robust from a bleaching perspective, you might cause harm, you might cause damage to your sample. And particularly over the past few years, there's been a lot of work and a lot of papers have come out showing these effects and showing how even if you can still see your sample and you still image it, you might be doing harm to that sample. So there is this need to try and lower power into the sample in order to get more representative results. So this is just saying, some of the techniques which are very useful for super resolution, we sell most of these techniques. They can also cause harm for live cell experiments. And then another motivation behind trying to push this triangle of speed, resolution, and sensitivity is context. We're used to seeing samples like I show here, where we have cells trying to get the best resolution from them, and in this case from cochlear cells within an ear, mouse cochlea. But it's more useful if you can capture this rich, high-resolution image, but also maintain the context of the whole ear. So you don't just want to get these high-resolution images, you want to get them with some level of scale. And we see over time that isn't just changing from an individual lab, but there is a general drive to going from the left hand of the picture shown here, where we look at individual cells, to trying to get to larger and larger model organisms. And again, organoids is something which has become a big trend and drive in the past few years, where we look at larger and larger things. Now, this is a point where I always find it interesting in PowerPoints, because of course, when I put an image up, we try and put it big enough that you can see the image, but without a scale bar, you kind of lose context here. This slide I'm showing you shows a handful of images that we've shown over the past few years in talks, and of course they all look nice, they all look lovely in their own right. If I present them in the actual scales relative to each other, you see that sometimes some of the high dynamic images, some of the very fast images, are very small relative to other things. So if we can improve the speed, if we can improve the sensitivity, we don't just capture what we want to see, but we can capture a bigger area of that. Now, how do we do that? The LSM 980 scanhead is really a feat of kind of love and engineering over the past few years, where we've looked at each and every component in the scanhead. We've tried to optimize that for peak efficiency. So very briefly explaining the scanhead, we have laser light coming in. Here we go on to narrow angle dichroics. Here we call it the twin gate because we have two of them for different laser lines. That sends light through the scanners into the microscope, and then the returning light comes back through the scanners, through those dichroics, through the pinhole, and then it's sent via a few mirrors, it's spectrally dispersed, and it's sent on to this multi-array detector we call the quasar. That's the main heart of the confocal. And over the years, we've looked at each and every component here, and we've tried to optimize it as much as we can to get the most sensitivity out. And some of that work is more obvious than others. One story I quite like is on the pinhole. So the pinhole, we kind of think it's a bit boring at times because from 1984, it's a hole. The pinhole on the 9 series actually came as a spin-off from one of our sister divisions. So microscopy is actually only about 10% of Zeiss. We have other divisions making other things, you know, optics, consumer optics, cinema lenses. And one of our biggest divisions is lithography, where we make these massive multi-million euro lenses you see on the left here for wavelengths down to 13 nanometers. And yeah, you think microscopy is precise, these things have to be extremely precise. So when we were designing the pinhole that we use in the current generation systems, we were looking at how do we absolutely reproducibly move a pinhole so that you can open it, shut it, and keep it aligned for the lifetime of the system. And we use some of the R&D from the lithography division in order to ensure that our pinhole remains precise over the lifetime of the system. And that alone, you know, if you have a pinhole that's even a fraction of an area unit out, you can throw away 10, 20, 30% of your signal. Another example of how we've optimized the light path is looking at the scanning. So this is the duty cycle. So as you scan across the field, what ratio of your time is spent going from left to right, and what proportion of your time is going back to the beginning? In the case of the LSM9 family, we spend 85% of our time scanning and only 15% on the flyback. Other systems don't necessarily do that. A lot of systems use a sinusoid design where you spend 60% of your time scanning, 40% not scanning. And what this means is you have to put your light in a shorter period of time. So imagine if you're trying to give your cell a certain amount of light, and you have to turn that light up because you're getting it in a shorter time, you're more likely to cause damage to that cell. The other disadvantage of using a sinusoid scan is that you don't necessarily have the same pixel dwell across the field of view. So if you think about the experiments I mentioned earlier, such as FRAP, such as image correlation spectroscopy, those don't work as well if you've got uneven bleaching across the field of view. Now, I can go on and could talk for another half an hour about the beam path, but the punchline, I guess, of all of this is that the LSM9 series is based on an architecture which is the most sensitive beam path available. And there are numerous studies where you look at the signal to noise, where here we've got very similar architectures, so our previous 700, 800, where high signal to noise is better or low instrument noise is better here. And we see that even without the top-end gallium arsenide phosphide detectors, we perform extremely well. With gallium arsenide phosphide detectors, we have unbeatable sensitivity here. But you always want more. So that's where the Airyscan comes in. The Airyscan is an optional detector for the LSM9 series, where instead of sending the light to the internal quasar, we send that light to an external hexagonal gallium arsenide phosphide detector. And the reason we do that is to image not just collect the light, but actually image the light that comes back from our single point of excitation. Now, why do we do that? I'm going to give a brief explanation of the Airyscan now. So here we have an image at the bottom, which is a bunch of beads, 170 nanometer beads here, and we image that with a 1 area unit pinhole. Now imagine if you close your pinhole down and you move it about ever so slightly, and you took 3 images with the positions indicated here and you colored it. This is the image you would get. Now, if you look at the bead pattern on the bottom, now coming from the UK, I always think this looks like the UK and Ireland here. If you look at the island of Ireland over here, on the image on the right, you see 3 islands. Now, of course, there is only one island of Ireland, there is only one bead here, but by moving the pinhole, you see that from slightly different angles. In the case of the Airyscan, we utilize that by not just moving our pinhole, but we have a digital pinhole where we acquire all of these slightly moved pinholes simultaneously. And if we look at a ring of beads here, if you imagine what the first ring on that detector looks like, you see an image like this. So of course the beads are fixed. What's moving here is an animation of which of the 6 detectors was looking at the bead. If you average that signal together, you would get the image on the right. If you do that for the next ring out, you get a more pronounced movement because the digital elements are more offset. You would get the image on the right, a bit more blurry. And if you do that in the extreme for the outer edge, you would get an even blurrier image. So in summary here, basically, as you open up the digital pinhole, your image gets progressively blurrier because it has progressively less resolution in. But because each of these elements, we know where they are digitally, before we add them together, we can apply something called the shepherd sum, where we shift and add those together, and we get the images on the bottom. And what this allows us to do is really have the resolution of a small pinhole, but collecting all the light of a big pinhole. So just to show a kind of summary here, we have 3 images on the left, the confocal image, if you just acquired that simply, the one in the middle, if we shift those images, and the one on the right, we can apply some filtering because we have good knowledge of the 32 elements, and we can get roughly a doubling in resolution in X, Y, and Z. And again, just a throwback to the graphs I showed earlier, what this allows us to do with the Airyscan is really have the sectioning of a small pinhole, the resolution of a small pinhole, but the signal of a big pinhole. So this means our image gets improved from what we see here in terms of gallium arsenide to what we see here with Airyscan. Now, of course, even with a non-Airyscan system, you can always average more, you can put more laser power in, but here I have a comparison where the images on the left are taken with a PMT-based system, the images on the right are taken with Airyscan, and they're taken with single images, average of 4, relatively high power, it's still 0.2% on the top, and relatively low power, 0.02% on the bottom. And if I zoom into these microtubules here, you can see not only is the Airyscan always better, but if I compare, say, the image in the complete bottom right to a PMT average 4 here, what you tend to see is that the Airyscan, you can get away with comparable image quality with about 1/10 of the laser power. Now, that's nice, but we want more. We always want more, of course. So one way of doing that is to increase the speed of the Airyscan. On the 980, we have various different modes. For the purposes of time today, I'm just going to show one of these, which is 8Y mode. So here we shape the beam going in. We spread that light over 8 pixels instead of 1, and we image that onto the Airyscan detector. And by doing that, we can go much quicker because we sample 8 pixels at a time. We also have the benefit in that by spreading the light over those 8 pixels, the illumination is gentler. So again, you get a reduction in phototoxicity and bleaching. Now, why is that important? Here is an example where there was a group looking at cardiomyocyte tubules. On the top, they had a resonance scanner and they were looking at the buckling, and the image quantification was hard. They couldn't really see the buckling, and if they increased the laser power, the cardiomyocytes stopped beating. On the bottom right, here we have an image taken with the Airyscan Fast, which is our previous generation of Airyscan, where we were able to image quicker with higher signal to noise and without killing the cardiomyocytes. If I move on to today's system, so to the 980, the group didn't really need more speed, but they were able to image the same sample with a larger field of view. So if you remember what I was saying about context, the larger a field of view, the better data you can capture. Now, I'm going to put a table up here. I'm only going to put it briefly. It is in the brochure, which I'll refer to later, but just to give you an idea, If we run the 980 at 512 by 512 pixels in super res mode, we can go up to something like 47 frames per second. And depending on whether you want to go, it all depends on what resolution and speed you need in terms of the performance and so on you can get out. But numbers is one thing, real life is another. So I wanted to show you an example of what this really means. Here is an example of a neuronal sample, which was imaged on another system where we acquired an image of about 250 by 250 by 34 microns over an hour. And we were able to improve that image using some deconvolution to try and look at these neurons. We imaged the same sample, but with about twice the field of view in terms of X and Y on the Airyscan using this 8Y multiplex mode. We're able to do that in just 4 minutes. And that's 15 times faster, but with a much bigger volume here and with 2 channels. And if we zoom in, you see that not only are we much quicker using the Airyscan approach, we're able to get better resolution in terms of resolving the dendritic spines. So sometimes it's not just about the speed, but it's about usable speed. One last example here I will show is, didn't autoplay. So this is an example, this was taken on the LSM 900, but this is an example of cells that are treated with a cytotoxic drug. And then we image them with Airyscan over a period of 20 hours. And after the drug is removed, the cells recover quite nicely. And you see that the imaging does them no harm. So up to now, we have confocals. They're very, very powerful. They've got better and better with performance over time. So happy days, everything is nice until the systems become so complicated, you can't always necessarily replicate your experiments. So in the last part of the talk, I'm just going to talk about reproducibility in LSM. So Nature's been highlighting it can be very hard to reproduce your experiments, not just other people's, even your own experiments here. So we want to try and help people do that. There are different parts of your image acquisition, from the hardware side, acquisition, running the systems, and even analyzing your data. And all of these are areas where you may introduce variation. So what we want to do at Zeiss is provide tools so that you can do this in a reproducible way. Now, in terms of the systems themselves, we partly do that by putting in very good engineering, like I showed technology from the lithography lenses, so that you don't need to keep an eye so often, or the systems maintain alignments over time. But of course, real life things might shift. So we have things like calibration objectives where you can check the system. We have automated ways, system maintenance tool to do that. And even more latterly, we have things like predictive maintenance, where the microscopes, if you're happy to allow it, will check on their system. They will let your local service department know, not just when something goes wrong, but prior to it going wrong. If it senses the laser power is on its way down, you can get your engineers out in advance. Moving on though, what I want to concentrate on for the last few minutes is looking at the software side of things. So software is really becoming the driving force of confocals and microscopes in general. We interact with microscopes via the computer, via the PC. And where things have changed is we used to have this chain in the middle of, you would operate the microscope, take an image, process it, and analyze it. Now, more and more, we're adapting and we're feeding back the results of this to change and improve the earlier steps, and we're trying to tie the dots between these images. So I'm going to show a few tools and a few things we've invented to help you do that. The first has only been launched last week. This is something called AI Sample Finder. This is the idea that one of the first things that holds people up is really just finding the right area of the sample, finding focus, that first 5 minutes where you come to a system and you want to be able to get on and get working. So AI Sample Finder is a combination of LEDs here and some detection and some very smart software. The way it works, well, is you put the slide on, the system automatically, using the LEDs, creates a dark field composite contrast image, and it will automatically load that sample, it will focus it, it will identify, do you have a multi-well plate, do you have a dish, do you have a slide? It will mark that, it will work out what on your slide is maybe dirt or contaminant, what may be of interest, and it will do all of that with artificial intelligence in less time than it's taken me to explain this to you. It will then transfer the coordinates over to ZEN, and it will transfer the coordinates not just so you can see where you are, but so that if you want to acquire, you can hit go and it will acquire that. There is also a form of coherence contrast, so whatever tissue you have, you will get some kind of contextual contrast. So this is very, very good for shaving that first 5 minutes on a system down to 1 or 2 minutes. And it's also very good in a world of COVID, where we might not necessarily even go into the labs these days. We can send someone in to load the sample, and we can work from home on the systems now. The next thing we've done in order to make systems easier to work with and to get more data is Experiment Designer. This is where instead of just doing one experiment at a time, you can do multiple experiments. You can say, I want to run a time series in one region, run a time series in another region, plus do more complex things like a contextual overview. So here is an example of a zebrafish where the contextual overview is taken with camera. That's taken for the whole zebrafish. But then two particular regions where you want more detail. This is looking at I think the neuromast lateral line. We're able to zoom in and use the Airyscan to run time series in each of these areas, but run that at the same time. Here we go. I hope you can see this. Okay, so yeah. Experiment Designer lets you not just run one experiment, but run experiments in multiple positions over time. Let's see if PowerPoint's happy still. Okay. Now, it's all very nice having multiple experiments at multiple time. What matters is sometimes the context. So ZEN Connect is a piece of software where if you take images with your motorized calibrated stage, you can keep all of those in context to each other. And that's useful if you want to come back at a later date or even just see how things relate to each other. So this is an example of Drosophila taken from different areas, and we can log all of those in a collaborative project using context. Now, this software isn't just useful within one microscope, it can be used cross microscope. So here I have an example where we use Airyscan, which is a wide field slide scanner, to quickly screen a load of samples, but then you can load that data, you can take images on your LSMs, and you can correlate the information together. And you can do that across the whole Zeiss portfolio. So Zeiss is one of the microscope company with the widest array of microscopes, all the way from X-rays for looking at really, really large samples down to helium ion microscopes. This is just an example where we take an image with a structural illumination super resolution system and with an EM, and we can correlate all of those together in context. But as ever, we want more. And this is where guided acquisition comes in. So Experiment Designer is fine, where you say, I want to look at do an experiment in one place and another place and another place, but at some point you want to automate that. Guided acquisition lets us do that in a way where you take images with an overview scan, you work out what you want to see with image analysis, and you come in. So I'm just going to show you a brief movie here. So this is a slide of Drosophila, and here what the customer was interested in is not screening all of the Drosophila because that was going to take too long, but just looking at a few. So they take an overview image, they run a form of an image analysis, and from that they work out which 20% of the Drosophila have the right expression. and they can then move to those and do a detailed scan of just the Drosophila of interest. Guided acquisition is the way in which they do that, and ZEN Connect allows them to keep the context here. So here you see a kind of crude overview, but each of the Drosophila marked with a box here, we're able to image with the Airyscan in high resolution. So this is a way of getting the most data out of your system, but in a sensible timescale. Now, the key to this, the key to running guided acquisition is having that processing step where you can work out what you want to see, which Drosophila are of interest. And that's where segmentation comes in. So with the systems being as quick and sensitive as they are, anybody can get good images these days, and we have terabytes and terabytes of data, but segmenting is the problem. Not everyone can do segmentation, not everyone can easily set this up. And so what we wanted to do with Zeiss is introduce a way in which the average PhD student can come in and run this kind of segmentation and guided acquisition. So what is IntelliSys? IntelliSys is a way of taking your raw images, training them, so telling the microscope, this is what I'm interested in, this is what I'm not. The system will then work out some features, it will train the algorithms, and it can then apply that not just on your training subset, but on the image you actually want to look at. If I show you an example here, this is a scratch wound assay where the colors that look like Photoshop paint here, MS Paint here, are really that. They're just crude annotations. This is the wound, these are the dividing cells. And from that classifier, IntelliSys will automatically segment between these. So you can no longer have to spend hours in the lab highlighting each of your cells. And you can plot, for instance, here, scratch area, how many cells in the image. And IntelliSys is really flexible at doing this. It will work on pretty much any life science data, particularly those where you have poor contrast or hard contrast, such as in DIC, maybe in EM, but fluorescence, it works very well. So far, so good. Again, we want more. So the last thing I'm going to mention in my talk is APEER. APEER is a independent, let me put this correctly. APEER is a platform designed not just for Zeiss users, but for any users. It's a web-based platform where you can develop image processing. Now, why might you want to do that? In the example I just gave you with the scratch wound, you take your images in ZEN, you might do some form of image improvement, contrast enhancement using anything. It could be Python, Fiji, whatever your choice is. You might do segmentation, that could be with IntelliSys, it could be with something else. And then you might plot your data in Excel. And each and every step along the way, you might introduce error here. Your step might not be very reproducible. To share that with another collaborator might be hard. Well, with APEER, you can design these workflows, or you can find one that someone else has already designed online, and you can set this all up as a workflow which is reproducible. So I'll give the link to the website later, but this is a tool, and you can load these not just from Zeiss microscopes, but from any microscope. And this also lets us use some of the state-of-the-art processing. So I'm not going to go into full processing now because the talk's already long enough, but if I just highlight the picture in the middle here, this is an example with a sheep and a sheepdog. Now, to differentiate the sheepdog, which might be a black sheepdog and it might be white sheep, you can do that with semantic segmentation reasonably easily. Where it's a bit harder is to do instance segmentation. So yeah, as I say, IntelliSys is really semantic. It's the top one. It's enough for a student to get on with a project which might be tens or hundreds of hours. To do the bottom type is a bit more complicated and it needs more images. It's usually also quite hard. So just what I'm showing here is binary segmentation, very, very easy. This kind of segmentation, nuclear segmentation, IntelliSys allows, but this kind of segmentation where you're not just looking at what the cells are based on their look, but also where they are, the contextual information. That's more complicated. And that has been allowed within the Intellisys platform. And as of Monday this week, we've just launched this in the form of machine learning. So you can get this on APEER, free for everybody. And it will allow some of this really quite high-end image processing to take place. There are some pre-packaged ones that people have already done, so you can go over there and work on those together. So to wrap up and summarize my talk, in the first part, I pointed out confocal has really gone from just a way of removing out of focus light to becoming very, very dominant. And it really is dominant for two reasons. One, it strongly performs in terms of speed, sensitivity, resolution, but it also has this wealth of techniques, the F techniques I explained, the numerical techniques, a massive number of ways of getting not just an image, but a number out. These systems have become incredibly sensitive over time. Not only do you have a very sensitive beam path, but the Airyscan allows you to get that, push that to the limit of super resolution, the limit of sensitivity, and it will do that reliably. We have ways of keeping the systems there. And then finally, you have that the wealth of software techniques now to improve not just the time to get an image, but the quality of that image, the quality of the data, and the reproducibility. And so where we've gone through over time is 1956, Minsky came up with this idea of removing the light that was out of focus. Over time, we've used that to make machines that make more and more data, and now we need to swim through this sea of data, remove the data we don't need, and just look at the data we do. To do that, we're using artificial intelligence, a term that was coined in a workshop in Harvard in 1956 by a chap called Marvin Minsky. So yeah, Minsky was involved not just in the pinhole, but also in the AI we're now rolling out today. And with that, I will finish my talk, and I'm open for questions. I'm going to leave you with this screen where I'm just going to put a few links up. Of course, if you're interested in the LSMs, you have the links there. The new sample finder, there is the link there. Intellisys, APEER.com, I would encourage you to take a look there, whichever microscope manufacturer you have, because it uses bioformats to load data from any microscope. And for more videos on this, more detail on the Airyscan, more details on basics of confocal, I would encourage you to visit the Insight Hub at the bottom. Thank you.
So thank you very much, Chris, for your very interesting talk. And yes, as Chris just mentioned, please feel free to send us your questions via the question dashboard. I see a first question which already came in, Chris. So let's start with this one, I think. So with automation, etc., one can improve reproducibility, but does the results improve? So meaning, will they be closer to reality?
So let me get that right. So as the results improve, will they get closer to reality? Perhaps I misunderstood that. Can you?
Yes, maybe I can just let's read it out once more. So with the automation, etc., we can improve what you have referred to, reproducibility of our results, but does the results also improve with the better reproducibility? So meaning, will they get closer to reality? Will they get better when we also have a better reproducibility?
Well, of course, you would hope so. I mean, that's the strive and the goal. I mean, you go at that two ways. One is to get more data. I mean, there's the joke about you could get a paper in Nature in the 80s with one cell, and in the 90s with 10, and these days you need thousands. The more data you have, the closer an idea you will have that what you see is real. But the other way of coming at that are the techniques I mentioned in the first part of the talk. People don't just take an image these days, or they don't just do a FRET experiment. The best systems biology labs ask the same question in multiple ways. So if you can use FCS, if you can use RICS, if you can use these techniques to verify your data in 3 or 4 different ways, you have much greater confidence that what you see is real. Okay.
Oh, Okay, thank you. Chris, do you have more questions received? Maybe also beforehand.
I'm just looking. So there's one. What's the difference between the LSM 900 and 980? So the talk I kind of mainly focused on the 980. The 900 is the smaller system. The main differences are number of laser lines, number of detectors, and the 980 being the bigger system can do things like multiphoton, FCS, etc. But if you talk to us or you look at those brochures, we can guide you in the right way.
Okay, so there was just, I think, an add-on to the question we just had, Chris. Yeah, the attendee says, okay, I'll reformulate. Maybe all scientists would be wrong because they use all the same AI platform.
Yeah, I see what you mean. Yes, I mean if there is a fundamental mistake in the AI, then yeah, that would be wrong. It would be if you were running your data incorrectly. But what you're trying to do with the AI, and where this AI comes in particular use, is it's really about taking a process which is extremely manual and automating it. Yeah, when you look at these, the data that comes out of IntelliSys and now the APEER machine learning, you kind of know what you're looking at is right. What it's done is it's managed to go and look at 1,000 Drosophila and segment them and work out what's going on for 1,000 of them. So you've got 1,000 data points. You can verify it yourself. You can look at 3 or 4 of these Drosophila and you can work out, yes, that is the result I would have come to if I'd have spent 3 hours sat in front of it. But you've gone and done that for 1,000 of them. So of course, it's only as good as the kind of questions you're asking. You could come to the wrong conclusions, but this is about large N, large amounts of data, sifting through things you couldn't do unless you had a ream of PhD students doing that for you, and also removing some of the bias. You know, how many of you have looked down a microscope and gone, "Well, that one looks good, and that one looks a bit dead," and yeah. Yes, okay, you might want to completely exclude nonsense from your results, but having it in an automated way, you can also make your data analysis fairer.
Okay, thank you, Chris. So another question regarding the sample finder. Would the sample finder work on the LSM 880?
On the LSM 880, no, it's a newly released feature that works in ZEN Blue. So you need a microscope running ZEN Blue, which is the latest 9 series, and other wide fields. You should know if you're running ZEN Blue or not, but yeah, it does need that in order to work at the moment.
Okay, thank you. So good to know. So Chris, any other questions you have received from your side?
I'm trying to see, I'm struggling to open the window. So for all the technicalities, let me have a look. I don't think I see any more.
Yeah. So yeah, if there are no more questions coming in, so otherwise also let us know. You can also, of course, contact us as usually if there are any other questions coming up in the meantime. And I think then, Chris, we can close the session for today.
Okay.
And thank you very much for your talk, Chris. And we just received one other question.
Go on.
Is FLIM possible with LSM 980?
Yes. I mentioned that in the first part. We do FLIM either with VisFLIM or with NLO, and with a choice of electronics manufacturer and detector type, depending on what you're looking at.
Okay. All right. So last chance for your questions. And what was the reconstruction algorithm for the multi-pinhole?
The reconstruction algorithm. So we, with the multi-pinhole, with the Airyscan, we really do 2 things. The first is what's called the Shepherd sum. So this was a reasonably old idea, but basically the idea of just shifting the pinhole by the amount in which, relative to where it is on the detector. So if you know it's shifted over 0.2 area units to the right, you shift it over 0.2 area units to the left. So that's reasonably quick and easy. The second part is we apply a Wiener filter, which is a simple denoising filter, in order to improve the resolution a bit further. You can go further and get into deconvolution if you want to squeeze that a little bit more, but that's probably a bit much for today's talk, and it becomes a case of diminishing returns and being careful for artifacts. So the main algorithm we use in terms of the Shepard-Sum and Wiener filter, we know we can do artifact free and very, very quickly.
Good. If there are no more questions from your side, then thank you once again for the talk, for listening, as well for your attention. And of course, we are looking forward to welcome you at any other webinars we are offering during our series we just have started. And I wish you a very good day ahead, and stay safe and wherever you are in the moment. And goodbye, everybody.
Thank you very much. Bye-bye.
Bye.